Hi, I'm Beth Patel. I'm a development technologist at the Mayo Clinic Genomics Laboratory, and I'm really excited to share with you a prospective clinical utility study we recently performed using mate pair sequencing to evaluate acute myeloid leukemia, and specifically in complex karyotypes containing both 5q and 7q deletions. So I'll go over, I'll just do a brief overview of acute myeloid leukemia, go over some basics of mate pair sequencing, and then really hone in on that complex karyotype cohort with 5q and 7q deletions. I'll go over things that really caught our eye with this cohort and made us want to research it more. Then I'll also talk about genomic complexity and recurrent abnormalities that we detected in this group. Acute myeloid leukemia is the most common form of acute leukemia among adults, with the median age at diagnosis of 67 years. It's characterized by clonal expansion of myeloid precursors, and it's estimated to have 20,000 new cases per year in the US and 10,000 deaths. And I'm not going to go over this in much depth at all, but acute leukemias arise from rapidly proliferating immature hematopoietic cells or blast cells. And the diagnosis of acute leukemia is determined by the lineage of these precursor cells. So in acute myeloid leukemia, the rapidly progressing blast cells are of myeloid lineage. So at Mayo Clinic, we did a prospective clinical utility study, and we did this on over 100 Mayo Clinic AML patients. Most Mayo Clinic AML patients are offered conventional chromosome studies, an extensive FISH panel, and we have 3 different NGS panels that are relevant to hematologic malignancies. And so for these patients, we collected residual sample after clinical testing was completed. We extracted the DNA and ran mate pair sequencing. And this study was IRB approved. And for the sake of this presentation, we're really going to focus in on those patients with 5q deletions and or 7q deletions. So mate pair sequencing is a whole genome sequencing method of NGS that's optimized for detection of structural variants in the genome, including translocations, inversions, copy number variants, and loss of heterozygosity. It is not optimized for detection of small mutations, including point mutations, CNVs and inversions with footprints below 30 kb, and low-level abnormalities, including minimum residual disease testing. Also, as with many high-throughput assays, repetitive regions are often filtered informatically from analysis to prevent false positive findings or noise calls in the data. So to really understand mate pair sequencing and the power behind this assay, understanding the library prep is really important. So we'll start with a 2 to 5 kb fragment of DNA. We'll biotinylate the ends of that 2 to 5 kb fragment. We will circularize the DNA during library prep and then cleave it. And then that cleaved section containing biotin will progress further into the library prep and sequencing. And this fragment contains pieces of DNA from either end of your original fragment. And so through paired end sequencing, we would expect these reads to map 2 to 5 kb apart in a specific orientation. And when that happens, these are called concordant reads. Discordant fragments, on the other hand, are what happens when your paired end sequencing maps to either different chromosomes or to the same chromosomes but over 5 kb apart. And when this happens, it can be indicative of a structural variant. So back to our clinical utility study, we had some specific aims for this study. including evaluating the clinical value of mate pair sequencing compared to chromosomes in fish. This is actually an extension of our clinical validation. We have a live clinical test right now for mate pair sequencing focusing on AML abnormalities. We also wanted to observe novel findings in AML regarding genomic complexity and recurrent abnormalities. And then our future aims for this study are to make inferences between genomic features and patient outcomes. So our 5Q, 7Q co-deleted cases, we found something pretty striking in that, okay, great.